Hello, everybody. In this lecture, we're going to talk about mutations to DNA. So let's get right into it. By the end of this section, you should be able to describe how mutations contribute to evolution, describe the different types of mutation, including substitution, insertion, and deletion, and compare silent, missense, and nonsense mutations. Explain what a spontaneous mutation is, and discuss the primary contributors to spontaneous mutation, discuss various classes of mutagens, how they induce mutations, summarize the Ames test and how it works, explain what excision repair is and how it protects cells from mutations. Right. So there are three main categories of mutations. We have substitutions, insertions, and deletions. So they pretty much are self-explanatory. Substitution mutations occur when an incorrect nucleotide is added in place of a correct one. So when a T is needed, if you add a G, that would be a substitution mutation. An insertion mutation occurs when a cell adds one or more nucleotides to its genome sequence, and deletion mutation occurs when one or more nucleotides are removed from a genome sequence. In single-celled organisms, a mutation is more likely to be passed to daughter cells. So let's look at these a little bit further. All right, so here we have different options for what can happen with these mutations. So we can have silent mutations. And this would be a situation where you get a change in one of your nucleotides that result in the exact same wild type amino acid. So in this situation, we're trying to code for lysine using the codon AAA. All right, so the original DNA is TTT. When it's transcribed, it becomes AAA, and that codes for lysine. Now, if there's a mutation in that third T to become a cytosine, then the transcription is going to become AAG. Now, this is a silent mutation because it is going to give us lysine again. This is part of why the redundancy in that amino acid code is so important. Now, in this next situation, if we change the center nucleotide to a guanine, you now have ACA in your messenger RNA, and that codes for threonine. So when you change that amino acid, we get what's called a missense. Missense means that we still put an amino acid in there, but it's not the wild type amino acid. In the last situation, we changed the first T to an A, which turns into our mRNA to UAA, which codes for one of our three stop codons. We call this a nonsense mutation because it stops transcription. So here are a couple of things that can happen when you have insertions. So these are all just explanations of different substitutions. Here we have an insertion and a deletion. And so what happens when we add insertions or deletions is we cause what's known as a frame shift mutation. So it changes our reading frame. So here's our normal DNA, no mutation. We get our mRNA with our different codons, which code for our different amino acids. However, if we insert an adenine between this C and this T, then we shift our reading frame, and instead of getting this lysine with this TTT, we actually are going to have ATT, which gets transcribed to UAA, which is one of our stop codons. Now, alternatively, if we remove thymine here, then we're switching our frame shift. We're frame shifting again, and now instead of getting lysine, leucine, isoleucine, we get asparagine, leucine, tyrosine. So we've changed up our two of the 4 amino acids overall. And so the other thing you notice here is that there is no actual stop codon. So this is going to keep going, even though we only needed these 4 amino acids, until it finds a stop codon in the transcript or runs off the transcript. Now, mutations can be helpful, detrimental, or neutral, right? So those different types that we just kind of talked about were silent. We haven't talked about reversion yet. And then there's nonsense and missense and frame shift, which we've mentioned a little bit of those. So the silent mutations are those that I just talked about, where you actually get a change in the amino acid sequence, but it doesn't change the protein. Sorry, you get a change in the mRNA sequence, but it does not change the amino acid sequence. In a reversion mutation, you get a mutation where a base is substituted to correct a previous substitution. And so that one is going to either fix a previous silent mutation or fix a previous nonsense mutation or missense mutation. Now, our nonsense mutations are any modifications that result in a premature stop codon. And that's going to give you an incomplete protein. And in almost all situations, these are going to result in non-functional proteins, or at least reduced functional proteins. And so what we see here, instead of getting this glycine, we end up getting a stop codon because we have this change in this. We have this substituted base pair from a C to a T. All right. In a missense, we get a protein that is going to normally or probably be the same protein, although you could get a missense mutation that has a premature stop codon. If you get a premature stop codon, so you get a nonsense mutation, then obviously stops. But when we just get a regular missense that just changes our amino acid, then the whole protein can get made. And in some cases, that can result in a perfectly functional protein. In other cases, that can result in a non-functional protein. It really depends on the amino acid that gets changed out and which amino acid it's replaced with. So here's an example of a frame shift mutation in a sentence. So in this example sentence, we have 3-letter chunks that give "the boy ran for the toy." Now, if we insert a letter into the sequence, we change that to an unreadable chunk. So that becomes "the boy ran for Ito," right? So it went into some sort of demonic summoning. And I'm no longer speaking like clear words that are understandable. And so that's what happens when we cause a frame shift. So that can happen either by insertion or deletion. Now, if you do an insertion of 3, then you can kind of reduce the overall like negative effect. So here we have another insertion where we have ARA inserted, and we get "the boy era Bicken for the toy," right? So after we get this insertion in to the sentence, we get a little bit of part that makes no sense, and then it gets back on line. So these kinds of frame shifts can be less detrimental. So what we see in this frame shift mutation is when we go from this histidine, we get this threonine, and now we have this like histidine like string here. But because we cause this frame shift, we get a threonine and then a string of serines. And again, this is going to go on until it finds a new stop codon. the old stop codon isn't going to work anymore because it's not going to be in the reading frame anymore. So mutations can be spontaneous or induced. Spontaneous mutations just occur naturally due to DNA replication errors, which remember that's like 1 in 10 billion bases. This can introduce genetic variation even when you don't reproduce sexually, right? Because this can have tiny changes in the DNA that accumulate over time. Regardless, you produce what's called a mutant strain, and these are cells that carry the mutation, which are different from the wild type strain, which are non-mutated cells. Mutagens come in a couple of different formats. These are chemical, physical, or biological agents that increase mutation rates. So for chemical mutagens, it's going to be stuff like arsenic and asbestos and alcohol, tobacco smoke. Physical mutagens are going to be things like UV light, X-rays, gamma rays. And biological mutagens are going to be things like transposons and some oncoviruses. Carcinogens are mutagens that cause a rate of mutation that promotes the development of cancers. And so not all mutagens are carcinogens, but all carcinogens are mutagens. So this brings us to what is known as the Ames test. So in the Ames test, you have a histidine negative salmonella. So they aren't able to make histidine. So that's a mutant strain. And then you add them to a medium that doesn't have histidine in it. And then you incubate them. And you might see a few colonies, even though the salmonella doesn't have the ability to make histidine. You can sometimes get spontaneous mutations that result in histidine positive salmonella. This would be salmonella that can make their own histidine. And so you can test whether or not something is a mutagen using this test, this Ames test, because what you do is you take your control, which is what we just described, and you count how many spontaneous mutations you have. And then you do the whole experiment over, but in addition to the histidine negative salmonella, you add your mutagen. Then you're going to grow them on that same medium that doesn't have any histidine in it. And if it's a mutagen, then you will see more colonies that have the ability to make histidine than you would expect from spontaneous mutation, right? So there's going to be more than would be in your control. Now, their DNA is protected largely by the proofreading and repair mechanisms. DNA polymerase is going to proofread. RNA polymerase does not proofread. Regardless, even though they're proofreading and finding these errors and fixing them, some of them don't get caught. This leads to an error rate of about 1 in every 10 billion base pairs. Now, you can also get what's called excision repair. This is going to be another mechanism where you can fix damaged or mismatched nucleotides. And so excision repair is going to cut out damaged DNA, and then it's going to replace that damaged DNA that's been cut out with DNA polymerase 1. And then, of course, DNA polymerase 1, when it adds in the new bases, is going to leave a single-stranded nick, and ligase is going to come in and glue the backbone back together by making that phosphodiester bond. And so here's a big example of a time when excision repair is necessary. When you're exposed to UV light, it can produce what's called thymine dimers, where two thymines that are next to each other in the DNA will bind together, and they produce this little bulge in the DNA strand, and that prevents accurate replication. And so they'll just replicate that as a single, like DNA polymerase would just replicate that as a single nucleotide. And so you would end up having a one missed nucleotide. And so the DNA excision repair complex comes in and cuts out that bold. DNA polymerase 1 jumps on, and just like it would if it were a primer, like it was replacing the primer, comes in and it adds the new bases. And then it leaves you that little nick at the end, and DNA ligase glues it all back together. So you should now be able to describe mutations, how they contribute to evolution, uh, distinguish between these types of mutations. You should be able to explain the differences between spontaneous mutations and induced mutations, discuss mutagens, the Ames test, and excision repair machinery. That, I hope you enjoyed. I'll talk to you soon.